evening, everyone. I am Kanika Gupta, a PhD scholar from IIT Delhi.
Lucky. Good evening, ma'am.
Good evening. Good evening. Can you please, yeah, can you please switch? Yeah, thank you so much for your mic. As I was introducing myself, those people who were in last session must be knowing me.
Good evening, ma'am.
Good evening. Good evening to everyone. So, yes, I'm a PhD scholar from IIT Delhi and I am a PMRF scholar too. And I'll be taking this session for week two of for the course of Human Physiology. Okay, so whoever joined the last session must be knowing that in this session, we particularly do a revision of what was the lecture for the whole week, for the last week, and then we'll move forward in the direction of taking the doubts. Okay, and if anyone has any doubts in between, they can stop me and ask the doubt. There is no issue in that also. Okay, so yes, first I'll start with the revision part. We'll see what we did in the last week. Okay,
Good evening, ma'am.
good evening to everyone. Are we starting now?
Yes, ma'am.
Are my slides visible to you guys?
Yes, ma'am.
Okay, thank you. So yes, first of all, we should know what are cardiac muscles. So as we know, cardiac muscles are one of the three types of muscles, the initial three types of muscle that we have read. So in cardiac muscles, these are called as the cardiac myocytes, which are surrounded by a cell membrane called sarcolemma, as same as the skeletal muscles. They have a nucleus also, and the cells are packed with mitochondria as same as the skeletal muscle to provide the steady supply of ATP. We all know that it requires ATP for different functions. So it has mitochondria also for the supply of ATP, and also again, as this is a muscle and it has to perform the function of contraction and relaxation, so it also has this actin and myosin filaments present in it. As you can see, it does had striations on these muscles. These striation indicates that this muscle also has, same as the skeletal muscle, the light and the dark bands, and it has the regulatory proteins, the troponin and tropomyosin also. So working of cardiac muscles is a bit different from the skeletal muscle. The structure is almost similar. Yes, there are some differences, like these muscles are involuntary muscles. The skeletal muscles were voluntary muscles. For example, we can move those muscles according to our will, but the cardiac muscles are involuntary muscles. The movement is not in our control. Okay, so yes, next thing we'll proceed with is, okay, as you can see, I should use a pointer if I can. Yeah, no worries. No one wants to join. It will keep on going meeting. Sorry for the delay, guys. Okay. I request you all, please try to join meeting by 6:10 p.m. so that we can move ahead in the session without any interruption. Okay. So yes, as I was telling you guys, in these cardiac muscles, if you can see, there are these small projection kind of structures present. These are actually the membrane of the cardiac myocyte. As I told you, these cardiac cells are called as the cardiac myocyte. Okay, so these striations are actually in connection through these desmosomes or gap junctions with the plasma membrane of the previous cell to make a connection between them. These connections are important for the conduction of the signal that will be generated in the cardiac cells, so that it can move in a rhythmic manner to next cell and the contraction can happen in a rhythmic manner. Okay, so these are called as the intercalated discs. Okay, so this is the intercalated disc, and the gap junctions, as I told you, allow the action potentials to spread between cardiac cells by permitting the passage of ions between the cells, producing depolarization of the heart muscle. Depolarization is actually the travel of action potential and change in the action potential, so that the cell can get a message that this is the time to contract. Okay, so yes, next is the excitation contraction coupling. If you guys have listened the lectures properly, and if you remember the skeletal muscle contraction part, so you guys must be familiar with the term T tubules. Okay, so here also we have T tubules, but here the T tubules are actually 5 times larger than the T tubules present in the skeletal muscles. In the skeletal muscles, the T tubules were thinner compared to the ones that are present here. These are 5 times larger. Apart from that, they also have these DHPRs. These DHPRs are again the calcium channels. These are again operated by the change in the voltage. And then when there is a signal, the calcium ions from outside enters through these DHPRs, which open by the change in the action potential of the plasma membrane, and the calcium ions from outside to inside enters here. The difference in the calcium ion concentration from outside to inside is nearly 1,000 folds, 1,000 to 10,000 folds. The calcium ion concentration outside the cell is huge. So these calcium ions rush into um, the this sarcomeres, and when they enter these cells, they change this. Here the ryanodine receptors are also present, but the main difference between the ryanodine receptors present here and the ryanodine receptors that were present in the skeletal muscle is that the ryanodine receptors here are not attached to the DHPR, as they were attached mechanically in case of the skeletal muscle. Here they are not attached with the DHPRs, and so here the ryanodine receptors are called as the ryanodine receptors 2. The ones that were present in the skeletal muscle were called as the ryanodine receptor 1, and here the ryanodine receptor 2 are present. So they will again undergo a change in their conformation by the presence of these calcium ion that enters from outside to inside the cell, which will make these ryanodine receptors open. And the sarcoplasmic reticulum, as you all know, that sarcoplasmic reticulum are actually the storehouse of the calcium ions present inside the cell. So when these ryanodine receptors undergo a change in conformation, so calcium ions present in the sarcoplasmic reticulum will come out from the sarcoplasmic reticulum and enter again into the sarcomere and further help in the contraction of the muscle. Here also again there are present the calcium pumps which uses ATP to move these calcium ions back into the sarcoplasmic reticulum. So again, the process is same. It will use an ATP to when the calcium ion concentration inside the cell increases to a level, then these pumps start working and it will move these calcium ions back into the sarcoplasmic reticulum, making them stored into the sarcoplasmic reticulum again. Now the thing here is that calcium ions, as we are saying, these are the ions. So ions can't be stored in a place for without affecting the charge that will be present obviously around the sarcoplasmic reticulum also. So there is this protein which is called as the calcisequestrine protein, which stores which helps to store this calcium ion in a state that it won't affect the charge around the sarcoplasmic reticulum, and it won't affect the stability of this sarcoplasmic reticulum. Okay, so calcisequestrine is the protein which helps to store this calcium ions inside the sarcoplasmic reticulum. Okay, let's see step by step what they are saying. That the action potential enters from the adjacent cell. As I told you, there are gap junctions present between in the intercalated disc, which will help in traveling of action potential from one cell to another cell. Okay. Next is the voltage gated calcium channels open, and this will lead to the calcium entering inside the cell. Then this calcium induces calcium release through ryanodine receptor 2. Okay, here as I told you, there are present ryanodine receptor 2. Then the local release causes calcium spark. The release of calcium from sarcoplasmic reticulum is called as the calcium spark. Okay, then the summed calcium sparks create a calcium signal. Now as the calciums are entering from outside also, and the calcium that are present in sarcoplasmic reticulum are also getting released inside the cell, so together they will create a calcium signal. Then this calcium ion again bind to troponin to initiate the contraction. You almost remember that the troponin here is actually helping in unmasking of the actin sites, the myosin binding sites on G actins, which will be visible after the binding of this calcium to troponin, which will help the tropomyosin to move from its position, that is actually masking the myosin binding site of the actin. So it will help in the visibility of these myosin binding sites. The myosin heads will bind to the actin and the contraction will happen. Now, the relaxation occurs when calcium unbinds from the troponin. It will lead to the again the masking of this myosin binding site on the G actin. So calcium is pumped back into the sarcoplasmic reticulum for storage. As I told you, calcium pumps are there on the membrane of sarcoplasmic reticulum. Calcium again, one thing, another thing that works here is, as I told you, calcium is entering from outside also inside the cell, and it is entering in a huge amount. So this calcium also must be sent outside of the cell. So for this, that works is another pump that pumps calcium outside in exchange of sodium. Okay, it brings sodium inside and takes calcium outside. This pump work in this way. And here also another thing that works here, because the sodium is entering inside, which is not a natural state. So the concentration of sodium must also be maintained. For that we have sodium potassium pumps that works tirelessly whole life. So these sodium potassium pump will move the sodium outside. Okay, and the sodium gradient is maintained, as I told you, by the sodium potassium ATPase pumps. So in this way the excitation contraction coupling will happen. Okay, so yes, this was the way. Just a moment, I guess someone wants to enter the meeting. There's no one. Okay, now there are admin. Just a moment, everyone. Okay, so next thing what we will do here is, yes, again there is, okay. So yes, now you guys must have seen this graph in the videos also. This is a very important graph to understand the contraction happening inside the cardiac muscle cells. So what happens is, generally the potential that is maintained inside the cell is minus 85 millivolts. Okay, why minus 85 millivolts? Because the positive charge outside is much higher than inside. So compared to that, the potential inside is measured as minus 85 millivolts. Okay, it is maintained as such. But when there's this action potential and there is a sudden entrance of the uh, calcium ions inside, when there is this action potential and the sodium enters inside the cell, causing a sudden increase in the membrane potential, and it reaches to nearly plus 20 millivolts. Okay, so it reaches to that, and then this change in action potential leads to the opening of the calcium channels also. So calcium ion also starts entering. So this plateau, you can, if you can see this, just on the point, so we can understand it better. Yes, this plateau, as you all can see, this plateau is because of the calcium ions that is constantly entering inside the cell, because of the change in action potential that is caused by the entrance of sodium ions inside the cell. Okay, and this calcium ion when enters inside the cell, it causes the calcium ion from sarcoplasmic reticulum to release also, leading to the calcium spark. Okay, so calcium permeability and inward flux increases, causing this plateau like situation. And when the conduction has happened and the calcium is again start going outside, So these calcium pumps, as I told you, are working in both ways, on the plasma membrane also and on the sarcoplasmic reticulum also, leading to the sudden decrease in the calcium ion concentration, so that the plateau is also decreasing and the action potential is again coming back to minus 85 millivolts. Okay, so this is the actually contraction, the sudden increase in the membrane potential to plus 20, and then the conduction is then this calcium then this plateau formation due to the increase in calcium ion concentration inside the cell. Okay, you can understand from this graph also that this sudden increase of the sodium ion concentration, and then again the sodium ion concentration, as the work of the sodium ion here is the change in the action potential. When it has happened, the calcium channels has opened, then it will suddenly decrease. It will decrease because the work has been done, the calcium channels has opened. Now it will suddenly decrease, and the influx of sodium ion will stop. It won't go beyond that, and the concentration, as you can see, it is decreasing. The influx of the influx of the sodium ion is decreasing and it is coming back to its normal. But due to this change in action potential, the conductance, the entrance of calcium ion inside the cell is increasing, causing this plateau like condition. And the calcium ion channels also, then after reaching a certain concentration, it will also start decreasing because of the pumps present inside the cell, causing a contraction inside the cell. And here the potassium ion also play a role in maintaining the permeability. The outward flux of the potassium ion will also start to bring to repolarize the cell and bring the action potential back to minus 85 millivolt, which will further then maintain to the concentration as it was through the help of the sodium potassium pumps, which will again maintain the sodium concentration and potassium concentration across the cell membrane. And in this way the contraction of the whole cardiac muscle happens, and it happens, as you all know, it happens in a tiny period of zero point 0.8 seconds. Okay, you all know one heartbeat is generally lasts zero point 0.8 seconds. Okay, so in this way we will see further also that generally 72, sorry, beats per minute is what is the normal condition for a heart. Okay, so yes, this was what it was. Now again we'll see why plateau. So there are fast sodium channels in the skeletal muscle. In cardiac muscles, there are fast sodium channels also, which open and closing in 10,000 of second, and slow calcium channels. As you guys must remember that when we were discussing the skeletal muscle also, the DHPR were called as the L type of calcium channels. This L type of calcium channels means long lasting calcium channels. These are the slow calcium channels. Okay, so here also these are called as the L type of calcium ion channels, same as the DHPRs. These are slower to open, then stay open for a longer time, and these cause the prolonged depolarization and this plateau. As I told you, this plateau is because of the calcium ions, the influx of the calcium ions, and these calcium ions play an important role in the muscle contraction. Just a moment, I guess. What is happening? Oh, it is not admitting. Okay, okay. So yes, Kiran, are you able to enter the meeting? Yeah, yes, I've admitted you. So yes, then what happens is this causes the prolonged depolarization and the plateau. These calcium ions play an important role in muscle contraction. Okay. Secondly, another peculiarity of the cardiac muscle is that the potassium permeability is greatly decreased. That helps to maintain the plateau. Okay, as I told you, because potassium ions further help in the maintenance of the action potential back to normal. so their permeability is decreased to keep the cell in the depolarized state. Okay, when the slow calcium channels close, causing the end of the plateau, and the potassium permeability increases rapidly. Okay, now calcium channels has closed, plateau has ended. Now the potassium channels permeability, potassium ions permeability will increase, helping to restore the repolarization. Okay, repolarization will happen, and then again the sodium potassium pumps will help, and other channels that are present on the plasma membrane will help to restore the state back to the polarized state for the cell. Okay, so as I told you, rhinodine receptor 1 are primarily expressed in skeletal muscles, and rhinodine receptor 2 are primarily expressed in myocardium या फिर heart muscles. As I told you, cardiac muscles. Okay. Okay, rhinodine receptors mediate the, as I told you earlier, also mediate the release of calcium ions from the sarcoplasmic reticulum, an essential step in the muscle contraction. In skeletal muscle, it is thought that activation occurs by a physical coupling as they are mechanically attached to DHPRs in case of skeletal muscles, but in case of cardiac muscle, it happens by the calcium ions that are entering from outside to inside of the cell. So, in cardiac muscle, the primary mechanism is calcium induced calcium release. You guys should remember this thing, that in case of cardiac muscles, the opening of rhinodine receptors, it is not a mechanical process, but it is by the induction through calcium ion only. Okay, calcium ion will induce the opening of rhinodine receptor and release of calcium ions. Okay, so this is an important thing you must remember. uh, and I told you there is an exchanger on the plasma membrane which will exchange calcium ions in exchange of sodium ions. So sodium ions enter, three sodium ion enter inside the cell in exchange of one calcium ion going outside, which is further then maintained by the sodium potassium pumps. Okay. Then the next thing is L-type calcium channels. As I told you earlier, these are the long-lasting slow calcium channels. One thing here that I want your attention towards is the intracellular calcium ion concentration in cardiac myocytes at rest is 0.0001 millimolar. As I told you, 1000 to 10,000 folds lower than outside. Inside the myocytes, the concentration of calcium ion is 1000 to 10,000 folds lesser than outside. In extracellular fluid, its concentration is 1.2 millimolars, while inside it is 0.0001 millimolar. Okay. In addition to calcium ions that are released into the sarcoplasm from sarcoplasmic reticulum, a large quantity of extra calcium ions also diffuse from the T tubules. As I told you, DHPRs will open, so these calcium ions from outside will diffuse inside the in the myocytes through the T tubules during action potential. And indeed, without this extra calcium from T tubules, the strength of cardiac muscle contraction would be reduced considerably, because there's a difference of 1000 to 10,000 folds in the calcium ion concentration from inside to outside. So the strength of the contraction would be very, very low if these calcium ions from outside won't enter inside the cell. Okay. So the sarcoplasmic reticulum of the cardiac muscles is less well developed than that of the skeletal muscle. Another reason of low calcium ion concentration of the requirement of calcium ions from outside is that the sarcoplasmic reticulum of cardiac muscle is less well developed than that of the skeletal muscle and does not store enough calcium to provide full contraction. Okay, so these are some important points that you guys must remember regarding the cardiac muscles. Another thing is the T tubules of the cardiac muscles have a diameter, as I told you earlier also, it is 5 times larger than the skeletal muscle T tubules, which means a volume of 25 times greater than the T tubules of the skeletal muscles. Okay. Also, inside the T tubules are a large quantity of mucopolysaccharides that are electronegatively charged and bind an abundant store of calcium ions, keeping these always available. Okay, so these are some important points that you must remember. Now, the next thing that happens here is this is a basic cardiac cycle you must have read in your like sixth or seventh class or eighth class somewhere. This is the basic blood flow. If you guys don't know it, no worries. I'll repeat it from the very basics. So what happens is in heart we have a right portion and a left portion. We have four chambers in the heart, as you all can see in this picture. We have four chambers: the right auricle, right ventricle, the left auricle, and the left ventricle. These are the four chambers inside the human heart, and the right part of the heart, in which includes the right auricle and the right ventricle. It This part is for the deoxygenated blood. Okay, this part of heart actually carries this deoxygenated blood, and the left portion of this heart, which includes the left auricle and the left ventricle, it will carry the oxygenated blood of the body. Okay. Now when we come to the functioning part, so there are two vena cavas. Vena cavas are veins actually. These two, just a moment guys, sorry. Yeah, where were we? The two vena cavas, the superior vena cava and the inferior vena cava. These are the two vena cavas that will bring deoxygenated blood from all over the body to this right auricle of the heart. Okay. Then through this right auricle, it has a tricuspid valve at the junction of an auricle to ventricle, right auricle to right ventricle. At its junction, it has a tricuspid valve which will help this blood to flow from right auricle to right ventricle. It opens and blood flows from right auricle to right ventricle, and then in the right ventricle there is this opening. Yes. Okay. So, as I told you earlier, also, yes, the deoxygenated blood that has been collected through these vena cavas to this right auricle will enter to this right ventricle. This right ventricle will then contract, and it has an opening of this pulmonary artery. Okay, if you can see this in blue, I'll show pointer and then this pulmonary artery. So when this right ventricle contracts, this pulmonary artery has a semilunar valve that will open due to the pressure, and this deoxygenated blood will enter into this pulmonary artery. Then this pulmonary artery will take this deoxygenated blood, as you can see. This pulmonary artery will take this deoxygenated blood to lungs, and in lungs we have alveoli where the exchange of this carbon dioxide with oxygen will happen. Excuse me, someone raised the hand. Yes, Sumi, you have a question. You raised a hand. Do you have a question, or should I proceed?
Yes, ma'am, you will proceed.
Okay. So, yes, as I told you, the deoxygenated blood through these pulmonary arteries will enter the lungs where there are present alveoli through which the blood will exchange, and the deoxygenated blood will now be oxygenated. Okay. So this deoxygenated blood will be converted into the oxygenated blood. Basically, the exchange of oxygen through carbon with carbon dioxide will happen here, and the oxygen will enter to this deoxygenated blood, making it oxygenated. Now, from these lungs, the through pulmonary veins will enter this left auricle, or we would say now the blood will enter to the left part of the heart, and it will enter to this left auricle. In this left auricle, the all of the oxygenated blood will come and get collected, and we have a bicuspid wall on the junction of this right auricle and right ventricle. So this bicuspid wall will open, and the blood will enter from this auricle to this ventricle. Again, the ventricle will contract, it will generate some kind of pressure, which will lead to the opening of semilunar valves present in the aorta. Here we have systemic aorta. Okay, it is an artery. So this systemic aorta, the opening of this systemic aorta will open, and the oxygenated blood will enter to this systemic aorta, making it to take the blood to different parts of the body and distribute it there, where this oxygenated blood will further supply the oxygen. Okay, so this is the basic functioning of heart. This is how the whole cycle of the oxygenated and oxygenated blood works. Now, there are two circuits here. One is pulmonary circuit, another is systemic circuit. The circuit which takes the which brings the deoxygenated blood to heart, and then this deoxygenated blood is taken to lungs, is called as the pulmonary circuit. Again, I'm telling you, the deoxygenated blood, when it is collected into the heart and through heart it reaches to the lungs, this whole cycle of the deoxygenated blood carrying and taking it to the lungs is called as the pulmonary circuit. And bringing this oxygenated blood from lungs to heart and through heart to different organs of the body, this circuit is called as the systemic circuit. Okay, so these are the two circuits present in the these are the two circuits that are formed by the heart in the body. This is the pulmonary circuit and the systemic circuit. I hope this basic cycle of the oxygenated deoxygenated blood supply to the heart is clear to everyone. If you guys have any doubt, you can ask me. You can ask me now, you can ask me later. Totally your choice, no issues at all. So this is how the blood is deoxygenated, the deoxygenated blood is carried to lungs, and then oxygenated blood is brought to different parts of the body. Okay, now we'll move forward, but I'll just allow this. Okay, I hope आप सबको समझ आ रहा है. There is any problem, you can ask me. Okay, so next thing, as I told you, the heart as pump. This is just the repetition of what I have told you guys now, but there are some terms that you must be knowing, so I'll tell you the terms. Okay, right atrium, you guys know. The valve, as I told you, is the tricuspid valve. It is also called as the right atrioventricular valve. Okay, right atrioventricular valve. Why atrioventricular? Because it is present at the junction of atria and ventricle. Atrium and ventricle. Okay. Okay. The You guys know the pulmonary vein and the left atrium, as I told you. Same as right atrioventricular valve, we have a left atrioventricular valve, a bicuspid valve. Okay, it is also called as the bicuspid valve. Uh, Apart from that, I have told you everything. There is nothing new in this. If still there is some confusion, you can ask me. We will move forward now. Now, this is a very important image, guys. This is just to understand the change in pressures at different points in the heart. So, first we'll see the change in the. We'll consider this for left ventricle. Okay. So, what happens here is initially when the blood is collected inside the atrium. Okay, as blood is coming from lungs, there is no such some kind of pressure point in between, or there is no contraction or relaxation in between when the blood is traveling from lungs to left atrium. So the pressure on the blood, so the pressure inside the atrium is very low. It is very low, as you can see. It the mark is below. Just okay. So as I told you, the pressure inside the atrium is very low. Same goes for ventricle, as there is no contraction happening at some point of time. So the pressure inside is very low. Okay. But when the blood is collected inside the atrium, and through this atrium, when the blood is getting collected in the atrium, it is filling the atrium. Then at some point of time, the bicuspid valve will open, and the blood from the atrium will flow from atrium to ventricle. Now what will happen is, as I told you initially, the pressure inside the ventricle is very low, but if you can see a little bit of shift in the atrial blood pressure due to collection of blood, so the pressure will be a bit higher in atrium than the ventricle, which will lead to a pressure difference between the atrium and the ventricle, leading to the opening of this bicuspid valve, and the blood will flow from atrium to ventricles. And then just went up. Oh, no, sir. Okay. No, someone is there. So, this valve will open and blood will enter into the ventricles. Now, after entering into the ventricles, the blood is getting collected inside the ventricle. When blood is getting collected inside the ventricle, there is this point when the blood is actually filled inside the ventricle. The amount that enters, the maximum amount that enters inside the left ventricle at some point of time, at a point of time, is 110 to 120 ml. Okay, this is the amount which enters the ventricle from from the left atrium. Now, when the blood is collected, the bicuspid valves are closed and the semilunar valves are also closed. This is the situation which we called as, if you can see, isovolumic contraction. Okay, this is the situation of isovolumic contraction. Both the valves, the bicuspid and the semilunar valves, both are closed. Now the pressure is, now the left ventricle will start contracting. It will contract till the point the pressure inside the ventricle reaches 120. Now one thing happens here is, when the pressure is increasing, it is increasing. When the pressure reaches 80 mm of Hg, the semilunar valves open. The semilunar valves that are present on the systemic aorta, the semilunar valves open, and the blood and the pressure inside the ventricle is increasing, and it reaches 120. When it reaches 120 and semilunar valves are open, so the blood from ventricle is ejected inside the systemic aorta, and this phase is called as the ventricular ejection. Okay, so this phase is called as ventricular ejection, and blood is entered inside the systemic aorta. Now, it the pressure inside the ventricle is slowly starts decreasing, bringing it back. When it brings back to minus, sorry, not minus, it brings back to 80 mm of Hg. The semilunar walls, sorry, when this uh, blood pressure is decreasing inside the ventricle, the semilunar valves has closed. Now the obviously the ventricle must come back to its relaxed state, so it will to its relaxed state. So uh, the blood that has flowed inside the systemic aorta, now as there was already blood present in systemic aorta, and the blood that has flown from ventricles to the systemic aorta has 120 mm of Hg, so the blood pressure inside the systemic aorta is maintained to nearly 100 mm of Hg. So this is called as the this dichrotic notch. This sudden drop in the blood pressure, but the blood must flow into the different parts of the body, so the pressure is maintained in the systemic aorta, and blood is then flows inside the systemic aorta and distributed to different parts of the blood, and inside the ventricle as it has started relaxing, so it will come back to its diastolic position. So the blood pressure will again drop to low as it was before. So this is how the blood pressure changes, and the blood is ejaculated in the inside the systemic aorta. The blood pressure inside the systemic aorta must remain minimum to 80 mm of Hg, so that it can be distributed to different parts of the body. It has to reach the highest point to the brain also, and to the lowest part, that is to the legs also. The blood pressure inside the systemic aorta must be maintained. Okay. And the atrial blood pressure will move as such, it will maintained as low, because it has to just uh, move forward the blood from atrium to ventricle. Okay. So this is how this happens. And there is this phase of isovolumetric relaxation. When the ventricles are relaxing, the semilunar valves are also closed, and the ventricles are relaxing, so this is the phase of isovolumetric relaxation. And then again the next cycle starts when the atrium again gets the blood, and this is how the whole cycle proceeds. Okay. So this was the cycle, the contraction cycle. Now there are some these terms which are very important for you guys to know. The end diastolic volume, as I told you, when the ventricle is fully relaxed. So the volume, the end volume of the left ventricle is nearly 110 to 120 ml. Okay. And the end systolic volume, that is after the powerful ventricular ejaculation, when it has sent the maximum blood through systemic aorta to different parts of the blood, different parts of the body. So the volume that is left inside the ventricle, as whole of the blood can't enter inside the systemic aorta, some blood is left inside the ventricle, and this is nearly 40 to 50 ml. Okay, this is 40 to 50 ml. So the stroke volume is the volume of the blood that has entered inside the systemic aorta when ventricles are ejaculating, or when ventricles are actually contracting and the blood is entering inside the systemic aorta. It is nearly 70 ml in the resting stage. It can increase up to nearly 100, when the person is in exercising state or something. But yes, it is nearly 70 ml in resting state. Okay. And you guys must know the cardiac output is calculated in a way that it
Ma'am,
yes.
ma'am, what is systolic volume, ma'am?
Sorry.
Systolic volume, ma'am. End of
Systolic.
systolic volume.
Okay, as I told you, I'm telling you. So what is systolic volume is, systole is the condition in which the ventricles are contracting. Okay. And when the ventricles are contracting, so it will increase the pressure. As I told you, the pressure will increase up to 120 mm of Hg. So
Yes,
this
ma'am.
increase of pressure will lead to the ejaculation of blood from ventricles to systemic aorta. But when ventricles are contracting, as I told you, the end diastolic volume is 110 to 120 ml. But this whole volume of blood can't enter into the systemic aorta when ventricles are contracting. So some part of the blood is also left inside the ventricle after the systole. The term is end systolic volume. So after the end of the systole, the blood that is left inside the ventricle is that volume. So when this ventricle ejaculation has happened, the maximum amount of blood that enters in a resting stage in the systemic aorta is the stroke volume, that is nearly 70 ml. So this is that amount of blood that is left inside the ventricles after a powerful ejaculation of blood inside the systemic aorta. Okay. So end systolic volume is
Okay,
the
ma'am.
volume that is left inside the ventricles after it has ejaculated maximum amount of blood inside the systemic aorta. Got it?
Yes, ma'am.
Okay. So yes, the cardiac output is calculated in a way, the if you will multiply this strokes volume to the number of heartbeats per minute, it will tell you the cardiac output. Basically, the amount of blood that heart has uh, sent to the whole body in a minute. Okay, so here the strokes volume is 70, and when you multiply this 70 with 72 beats per minute, 72 beats per minute is the resting stage of heart, in which in 1 minute it will beat for 72 minutes, which is a normal condition, which is said to be. So when you will multiply this, you will get it around 5 liters. Okay, the amount is around 5 liters. So it is said that the
Ma'am,
cardiac output is, yes.
ma'am, when that blood will enter into systemic, ma'am, systemic aorta, remaining blood 40 to 50 ml blood,
Actually, this is the amount which stays in the ventricle. This is the amount of the blood
nothing will
that
happen,
stays in
ma'am.
the ventricle.
If it stays, nothing.
Sorry.
If it stayed, if it stays in the ventricle only, nothing will happen, ma'am.
No, 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 no. It is a very normal condition. If you can see the slides, there is an end diastolic volume. Okay, so this much amount of blood is already present in the ventricle after a powerful systole, and end diastolic volume is the when the ventricles are completely relaxed. So some amount of blood is entering from atrium to ventricles. So the whole amount of blood that is present at a resting stage at the end of a diastole in a ventricle is 110 to 120 ml, and after systole, when nearly 70 ml of blood has entered the systemic aorta, this much amount of blood is left inside the ventricle. Okay, there's not this is not
Okay,
a
ma'am.
problematic condition. This is the normal condition of heart.
Okay, ma'am. Thank you, ma'am.
You're welcome. Now next, this okay. So another thing, that is when the heart contracts strongly, the end systolic volume can be decreased to as little as 10 to 20 milliliters. This happens when we are in some kind of the exercise condition, or we are doing some kind of a strenuous activity where the body needs the extra supply of oxygen. It requires extra energy. So in that situation, heart contracts more, with more power, leading to the decrease in the end systolic volume to 10 to 20 milliliters. It happens in case of the rigorous activities, or in kind of in the situation of the strenuous activities. Okay. So conversely, when large amounts of blood flow into the ventricles during diastole, the ventricular and diastolic volumes can become as great as 150 to 180 milliliters. Okay, so the end diastolic volume can increase up to 150 to 180 milliliters, as I told you, in some situations like exercising. Okay, uh, so this increases. One thing that you guys must remember here is the amount of blood that will enter during the systemic circuit in the systemic aorta is the amount of blood that will enter into the right atrium, the deoxygenated blood that is entering into the right atrium. The amount that is entered the systemic aorta is the same amount of blood that will enter the right atrium also in that particular cycle. Okay, so the amount of blood that has entered the systemic aorta is equal to the amount that has entered in the right atrium, the deoxygenated blood. So in case of the exercising condition, the cardiac output also changes, and the amount of blood, the end diastolic volume, the systolic volume also changes. As I told you, cardiac output. So cardiac output is simply the amount of blood pumped by heart per minute. And necessarily, the cardiac output is the product of heart rate, that is around 72 beats per minute, which is the number of beats per minute, and the strokes volume, which is in resting condition is considered to be 70 ml per beat. Okay, and when you multiply this, it will come out to be around 5020 ml, that is nearly around 5 liters. And 5 liters is the total amount of blood that is present in a human body. Okay, someone wants to join the meeting. Why it is, what happened? meeting. What happened to the meeting? I'm so sorry, guys. I guess there was some problem in my laptop.
मैम आपका माइक बंद है।
Ma'am, your voice is not visible, ma'am.
Okay. Now my voice will be there. Actually, I guess there was some problem in my laptop, and by mistake I left the meeting. I'll just show you guys just a moment. Okay. Okay. Are my slides visible to you guys now? Any?
Yes, ma'am.
Yes, ma'am.
So as I told you, cardiac output is the multiplication of heart rate with the strokes volume, which is around 5 liters per minute. Okay. So now, next thing is the cardiac conduction system. The cardiac conduction system is a group of specialized cardiac muscle cells in the walls of heart that send signals to the heart muscle, causing it to contract. Okay, sorry. When we were reading about the excitation and coupling system, we read that how the cardiac muscle contract, but there must be some system involved which will send the signal constantly to these T tubules through these cardiac muscles that yes, you have to contract, you have to relax, and all this. And as I told you earlier, also these cardiac myocytes are not voluntary; these are involuntary. So there must be some automatic system that is sending the signal, and this is the cardiac conduction system. The main components of cardiac conduction systems are the SA node, the internodal tract, the AV node, bundle of His, bundle branches, and Purkinje fibers. Okay, SA node is also called as the pacemaker of heart. Okay, it is called as the pacemaker of the heart. It is a self we'll say self exciting, it is sending the signals by self excitation. It is not getting some signals from outside or something. It is self exciting. Okay, self generating the action potential. Mm, uh, the laptop is getting slow day by day. Admit. ठीक है जी. So as I was telling you guys, yes, uh, let's see their location. Achha, one thing you guys must remember is the signal in heart works in a rhythmic manner. It's not like when we saw in case of skeletal muscle, it was happening in milliseconds and it was happening for the whole muscle. But here it will happen in a rhythmic manner. First signal will go to some part of the heart, then it will move to the another part of the heart, and this way, in a rhythmic manner, it will travel in the whole heart, and the contraction will happen. Okay, so let's see the location. This is the SA node, this is the AV node, atrioventricular node, this is the sinoatrial node, this is the bundle of His. Okay, and this is the Purkinje fibers, guys. These are the Purkinje fibers. So this is the SA node, which is called as the pacemaker of the heart. Each cardiac cycle is initiated by the spontaneous generation of action potential in the SA node. Okay, and the action potential travels along the conducting system to AV node, and through this AV node, it uh, goes to this bundle, these branches, and then in the bundle of through this bundle of His, then further there is a delay. There is a delay by 1/10 second. Why there is a delay? So that there is no simultaneous contraction of atrium and ventricles. So there is this delay in the bundle of his, and after that, through this bundle of his, this the action potential travels to these Purkinje fibers and the ventricles will contract. Okay, so there is this delay to prevent the simultaneous contraction of atrium and ventricles. Okay, just a moment. Sorry, yeah. So specialized excited and conductive system of the heart, the normal rhythmical, as I told you, it works in a rhythm, not simultaneously but in a rhythm. The normal rhythmical impulse is generated in the sinus node, the SA node. It is then conducted over the internodal pathways. As I told you, these are the internodal pathways. These are the internodal pathways. Okay, so it is conducted over to the internodal pathways to AV node, in which the impulse is delayed before passing into the ventricles. The left and right bundle of Purkinje fibers conduct the impulse to the all parts of the ventricle. Okay, so Purkinje fibers will take this action potential to the to all parts of the ventricles, leading to the contraction of the ventricles. Sorry, the SA node is a small flattened strip of cardiac muscles located in the wall of the right atrium. Now, one point here is, as these SA node, AV node, or the bundle of his or the Purkinje fiber, whatever cells these are actually cardiac muscle cells. But the thing here is, these cells do not have to contract. The work here of these SA node, this AV node, all of these things involved in the conduction system is to make the action, to generate the action potential and let it travel to the different parts of the heart to maintain a rhythmic impulse and rhythmic contraction. So the amount of actin and myosin in these cardiac cells is comparatively lesser than other cardiac myocytes. Okay, so the fiber of this node have no contractile muscle filaments. They are saying no contractile muscle filaments, and these are actin and myosin. These are comparatively lesser, much lesser. It can be nil, but they are comparatively much lesser than the other cardiac myocytes. However, the fibers connect directly with the atrial muscle fiber, so that any action potential that begins in the SA node spreads immediately into the atrial muscle wall, so that the the contraction can happen strongly and simultaneously in case of the atrium. Okay, so this is the SA node, as I told you, the pacemaker that generates impulses. These are the internodal pathways. The impulse pulse of 0.1 second at the atrioventricle node, and the atrioventricle AV bundle connects the atria to ventricle. ठीक है, this AV bundle connects the atria to ventricle, so that and there are this bundle branches that conduct the impulse through the interventricular septum. This is this junction between two ventricles. This not junction actually. This is the septum which separates two ventricles, the right and the left ventricle. This is called as the interventricular septum. And then there is these Purkinje fibers which will further conduct the action potential and will help the ventricles to contract. And this is the discharge rate of action potential in SA node, AV node, and Purkinje fiber. As you can see in Purkinje fiber, it is much lower than SA node and AV node, so that the ventricles won't contract unnecessarily. They will contract when there is the time to contract. Okay, so this was what we have studied in the last week in the lecture. Till now, if anyone has any confusion in the lecture part, they can ask me questions. Otherwise, we'll start with the assignment. Anyone, if have any confusion, they can ask me. And also, I have brought some questions other than assignment for you guys. So we'll do those questions also. It will help you in revision also, and it will help you to remember the important points also. So yes, any confusion till now can ask me, any problem. No?
No, ma'am.
Okay, then I'll proceed with this assignment part. I hope you all were able to solve the assignment. Let's see. Okay, what is this thing which is marked as 1? Can anyone answer this? This band. You have read this structure in skeletal muscles. So you must remember what is this band called? The one that is marked as 1.
And A band.
Yes, it is A band. And the one which is marked as 2, it is
Z line.
Z line. Z line, Z disc, anything you can say. So, the one that is marked as 3 is
Sarcomere.
not sarcomeres. See properly. The one that is marked as 3, it is the I band.
I
IBAN,
band.
There
ma'am.
is no
I
voice
band.
in here. It is only the axis of the Z disc. So it is the I band. Okay. And the one that is marked as fourth is this is sarcomere because it covers
Sarcomere.
the whole cell. Okay. The whole unit from one Z disc to another Z disc. Okay. So I hope there was
Yes,
no
ma'am.
problem in this question in solving this question. Okay. Now, what is this thing that is marked as one in this picture?
T
Intrathecal.
tubule, ma'am.
Yeah.
T tubule, ma'am.
No, it is not T tubule. See properly, it is a junction between two cells.
Okay,
Yeah,
sarcoplasm.
yeah. So it is
Z, Z line.
no Z line. We are not talking.
Intracalated disc.
Yes, it is intracalated disc because this is a junction
Intercalated.
between two cardiac muscle cells. So it is an intercalated disc. Okay, it is not Z line or something. It is देखो यहाँ पे desmosomes भी तो है ना. As I told you, desmosomes, gap junctions, these are present at intercalated disc. Okay, so remember this thing. This is intercalated disc. Okay, the one which is marked as 2 is
Sarcomere.
Sarcomere.
sarcomere, right? Very right. And the one which is marked as 3 is
Gap
Gap
junction.
junction channels.
gap junctions, right? And the one that is marked as fourth is
Plasma membrane.
plasma membrane. Perfect. You were able to solve the assignment, right? Okay. Now, the thing that is marked as 1. This point in the graph is,
Overshoot.
Depolarization, ma'am.
sorry?
Overshoot, ma'am.
Overshoot, ma'am.
Yes, it is overshoot. And this is the polarization of which muscle? This is the graph of contraction for which muscle, if you guys remember?
Skeletal muscle, ma'am.
Skeletal muscle, right? It is not for cardiac muscle, it is for skeletal muscle.
Skeleton.
Okay, the one that is marked as 2, this whole graph is for,
Repolarization, ma'am.
yes, it is for repolarization. The one that is marked as 3 is
Depolarization.
Depolarization.
depolarization, and the bar here is the white bar.
Millivolts.
Millivolts. Yes, this is the actually change in. This is actually showing the voltage, different voltages. Okay. Yes. Now here, this is the basic cycle as I told you earlier, pulmonary circuit and systemic circuit. So I hope you were able to answer it correctly. The thing that is marked as 1 is
Pulmonary vein.
Pulmonary artery, ma'am.
pulmonary vein. It is not a vein. It is an artery.
Artery,
Art-
ma'am. Artery.
Artery.
It
Artery.
is an artery. Okay. Now, the one which is marked as 2 is,
Left auricle, ma'am.
yes, it should be left auricle. I guess here the answer is wrong.
Left auricle, ma'am.
It is left auricle. Okay. The one that is marked as, sorry, the one that is marked as fourth is,
Pulmonary vein, ma'am.
no, it is left ventricle. I am asking about 4.
Left vein.
Left
4, okay.
ventricle.
Left ventricle, ma'am.
Left ventricle. And the one that is marked as 3 is,
Pulmonary artery.
yes, this is pulmonary artery. Sorry, I was wrong. The one who said it to be pulmonary vein was right. I got it is an artery, and the one that is bringing blood from lungs to heart is a vein. Okay, so this is pulmonary vein and this is a pulmonary artery. Okay, next thing. So this is the one which is marked as one is
Brahman's bundle.
Bachman's bundle. Yes, it
Backman's
is
bundle.
Bachman's bundle. The one which is marked as two is
Pathway.
Purkinje fibers, ma'am.
Purkinje fibers. Conduction pathway
Connection
is also
pathway.
called, this part is also called as conduction pathway, otherwise it is Purkinje fibers. Okay, the one which is marked as fourth is,
Ventricular nodule. Arterioventricular.
yes, atrioventricular node, AV node. And the one which
AV.
is marked as three is,
SA node.
yes, sinoatrial node. Okay, now hope there was no confusion in the assignment part. Let's move towards the questions that I have brought for you guys. This is just for your practice, nothing like no burden taken or something. So yes, the heart pacemaker is, these are four options: AV node, SA node, bundle of his, or Purkinje fibers. What is the heart
SA
pacemaker?
node, ma'am. SA
SA
node.
node, ma'am.
Yes, it is SA node. Okay, stroke volume is defined as,
The
Diastole of ventricle, ma'am.
stroke volume is defined as, again, read the question carefully and the options also.
Option B.
Volume of blood ejected.
Volume of blood that is ejected per
Ejected
beat.
per beat.
Yes, it is that. Okay.
ejected.
Then the next question is, the normal value of cardiac output in an adult at rest is approximately
5 liters, ma'am.
5 liters.
5 liters per minute. हमने अभी पढ़ा ही है, और आई होप आप लोगों ने ये मैथ्स भी करके देखा होगा. If you multiply 70 with 72, it will come out to be 5020 ml per minute. Okay, so it is generally 5 liters per minute. Okay, next question. Sorry, which ion primarily controls the plateau phase of the cardiac action potential?
Calcium.
Calcium.
Yes, it is calcium. बताया था मैंने कि calcium जो है, that is actually the ion due to which there is a plateau phase in the contraction of cardiac muscle. Next is baroreceptors that regulate blood pressure are located in which part? See the options and
Option
tell
B.
me.
Option B.
Carotid sinus and aortic artery. Okay, we saw in the week one lectures that there are baroreceptors present which are connected to which nerve?
Cardiac sinus and cardiac artery.
No, 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 no. I'm talking about which nerve is getting the signal from baroreceptors and leading it to the CNS. Which nerve was that?
Vagus nerve, ma'am.
Vagus nerve. Yes, it was vagus nerve.
Yes.
Okay. Okay, so the next question is, the primary function of hemoglobin in the cardiovascular system is, yes, if someone can
Option B.
answer this question.
Glucose transport.
Option A.
Option A, it's transport of oxygen. It is an protein to carry oxygen. Okay. Okay. So the main difference between arteries and veins is,
Venous carry. Arteries, venous are both blood vessels.
इन्होंने कहा है difference. What is the difference between arteries and veins? Okay, let's do
Oxygen.
one thing.
Venous carry oxygenated blood. Arteries carry deoxygenated blood.
Okay, okay.
Option. Option
No,
C.
ma'am.
Let's do one thing. Let's do one thing. You guys, in spite of answering like that, let other people also answer. Write your answers in the chat box and then I'll tell you whose answer is correct. Okay, let's do it that way so that everyone can participate. So write your answers in chat box. What is your answer for question 7? The question is, the main difference between arteries and veins is, just write the option number A, B, C, or D. Okay,
B.
your answer is B.
Yes, ma'am.
And And
Option
everyone,
is
other people, what is their answer?
option option C.
Write in the chat box, please. Write it in the chat box. Come on. Everyone, what is the answer? I hope सबने लिख दिया होगा. So answer is option C. Arteries have thicker walls. Okay. Those people who were saying it to be option B, let me tell you, the way it says veins carry oxygenated blood and arteries carry deoxygenated blood, which is totally false. We have seen that the veins carry deoxygenated blood and arteries carry oxygenated blood. It is just an exception that the pulmonary vein carries the oxygenated blood. Otherwise, all other veins carry always deoxygenated blood. Okay. So the main difference between arteries and veins is arteries have thicker walls. Okay. So yes.
Okay.
So the next question is, again, everyone will write the answer in chat box. No one will speak it loud. Okay. Let everyone participate. So the dichrotic notch in the arterial pressure waveform is due to what? We have four options: AV valve closure, semilunar valve closure, atrial contraction, and ventricular contraction. Tell me the answer. Everyone answer. वर्षा कह रही हैं ए. ओके, बाकी लोग. वर्षा इज सेइंग बाकी का कोई आंसर नहीं है, कोई आंसर नहीं करना चाहता इस क्वेश्चन को. ओके, वी आर गेटिंग सम मिक्स्ड आंसर्स. Everyone, please answer. We have many more questions. I hope you guys are enjoying this. It will help you remember things properly. So this is question 8. The dichrotic notch in the arterial pressure waveform is due to what?
Arterial contraction.
You write your answer in the chat box, Madhura.
Okay, ma'am.
Yeah. Okay, I'll tell you the answer, guys. This is due to semilunar valve closure. I told you the semilunar valves will close, and when the ventricle is start relaxing, the semilunar valve will close so that blood doesn't flow back from systemic aorta into the ventricles. As the ventricles, there is the drop in pressure in ventricles, so blood will try to flow from higher pressure to lower pressure. So to prevent this, semilunar valve will close. Okay, so this dichrotic notch. Now, if you remember what is a dichrotic notch, this is the dichrotic notch. This is the dichrotic notch. Okay, everyone. This is due to the closure of the semilunar valve. ठीक है? चलिए जी. Next question is, cardiac muscles differs from skeletal muscle because. You have these four options. Just tell me the answer. This is for question 9. Okay, we are getting different answers. Cardiac muscles. Let's read out the options. Cardiac muscle is voluntary, which we know it is not. It is involuntary. Okay. Then cardiac muscles lack sarcomere, which is not possible. Otherwise, how will it contract? Cardiac muscles has intercalated disc. This seems right. Okay, so whoever has written C, the answer is correct. Cardiac muscles has intercalated disc. And skeletal muscle has longer refractory period. It is not possible. As I told you, skeletal muscles take milliseconds to contract. Okay. So yes. Now let's move towards the next question. The next question is, it is question 10th. The blood vessel with the highest blood pressure is, चलिए इसका आंसर बताइए सब लोग. The blood vessel with highest blood pressure. इसका आंसर
Aorta, ma'am.
aorta. Yes, it is aorta because capillaries हमें पता है बहुत thin walled होती हैं. So there can't be much pressure in capillaries. Venues also don't have much pressure. And inferior vena cava is bringing the blood from different parts of the blood to heart, so it doesn't need much pressure. Okay, so it is aorta only. Okay, now the turn for question 11. During the isovolumetric contraction phase, what will happen?
Blood flows into the ventricles.
Blood flows into the ventricles. ठीक है, बाकी के ऑप्शंस भी देख लेते हैं. हाँ, बाकी के ऑप्शंस भी देख लेते हैं. सब लोग अपना आंसर दीजिए प्लीज इन द चैट बॉक्स. निशान, डू यू वांट टू से समथिंग?
मैम ये आइसोवॉल्यूमेट्रिक एक बार फिर से बता देंगे क्या?
Isovolumetric contraction.
यस।
Okay, let's move towards that then. So Nishan, the isovolumetric contraction is that phase in which the blood has filled inside the ventricles and the ventricles are now contracting. Okay, both the bicuspid valve and the semilunar valve are closed, and the blood has entered the ventricles and ventricles has started contracting, and the pressure inside the ventricles are increasing. So this is the situation of isovolumetric contraction. Here the walls, the main point here is the walls, the bicuspid and the semilunar wall, both are closed in this, the isovolumetric contraction situation. Okay.
यस।
हाँ जी, अब इस क्वेश्चन का आंसर करेंगे सब लोग. Isovolumetric contraction. During the isovolumetric contraction phase, answers please in the chat box, everyone. According to them, it is option C. Okay, Raj. ठीक है जी, तो इसका आंसर मैं बता देती हूँ. इट इज both AV and semilunar walls are closed. ठीक है, both AV and semilunar walls are closed. Let's move towards the next question. It is the longest phase of the cardiac cycle at rest is. बताइए भाई. The longest phase of cardiac cycle at rest. If you have seen that graph properly, I think you can answer this question. Everyone. Bhakta, option D. Rajatha, it's option D. Other people, please, all you guys also participate. It will help you to learn things better. Okay. So the answer to this question is, it is the diastolic phase. Diastolic phase is the longest phase of the cardiac cycle. Okay. So it is the diastolic phase. Okay. Now next, we'll move towards the next question. It is this. The force exerted by blood against vessel walls is called as.
Blood pressure.
Yes, it is blood pressure. Other people, what would you say? Please, guys, be active and try answering in chat box. It will help you guys to remember things better. Yeah, it is blood pressure only. Okay, if anyone had any confusion in the previous question, they can tell me. I'll show you the graph once again so I won't be confused about this longest phase of cardiac cycle. Just a moment. Okay, so if you can see, this is this phase, this diastolic phase, the relaxation phase, this atrial systole, और उसके बाद जो diastole. Actually, in this you can't see it clearly, but what happens is the diastolic phase is generally nearly zero point 0.4 seconds long, and the systolic phase is generally 0.3 seconds long. There must be somewhere mentioned inside the notes also, so you guys can see it from there only. There is not mentioned. This isn't mentioned here somewhere, but I'm telling you guys, the diastolic phase is nearly 0 0.4 seconds, and the systolic phase is nearly 0 0.3 seconds. Okay, if I remember correctly, if there is any change, I'll tell you in the next class. As far as I remember, it is this only. So I'll tell you guys if there is any change in that. Okay. Yes. So the next question, we have answered the question. The force exerted by blood against the vessel walls is called as the blood pressure. Next question is, which type of blood vessel is primarily involved in nutrient and gas exchange?
Capillaries, ma'am.
Navya, I request you, please write the answer in the chat box so that everyone can participate. Okay?
Okay, ma'am. Sorry.
No problem. No problem at all. So yes, everyone, what is your answer on this? Which type of blood vessel is primarily involved in nutrient and gas exchange? Nishant, your answer is A for this question.
Yes, ma'am.
Okay. Everyone else? So the answer to this question is option C, that is capillaries. Capillaries, because at the end, the capillaries is also is the one which is reaching to every organ, to every cell in the body. So it is actually the capillaries through which the gaseous and the nutrient exchange is happening. Okay. So the answer is capillaries. Next question is, which heart valve prevents backflow from the right ventricle to the right atrium? Read the question carefully. Which valve is this that prevents the backflow of blood from the right ventricle to the right atrium? Option B, option C, option C. Okay. So yes, the correct answer is the tricuspid valves. It is the tricuspid valves that will prevent the backflow of the blood from right atrium to right ventricle. It, why option B is wrong? Because you are saying aortic valve. Aortic valve तो right part में होती ही नहीं है. Aorta तो होती ही left part of the heart में. उसकी जो opening है, that is there in the left part of the heart, not in the right part of the heart. So B तो बिल्कुल ही गलत है. B is not the answer, and mitral valve is the name for another name for the bicuspid valves. Okay, so you can note it down. The mitral valve is the another name for bicuspid valves. So the answer is tricuspid valves. Okay, let's move towards the next question. Okay, so the coronary arteries arise from. Coronary arteries are those arteries which supply the blood to the heart. हार्ट की सेल्स को भी तो ब्लड चाहिए ना, ऐसा तो नहीं हो सकता कि वो सब पूरी बॉडी में ब्लड दे रहा है, उसको नहीं मिलेगा. So coronary arteries are those arteries which supply the blood to the heart, so to the heart cells. So the answer to this question, what is the answer to this question, everyone? Okay, so the coronary arteries arise from base of the aorta. Okay, क्योंकि इसको oxygenated blood देना है ना, so it arise from the base of aorta. Okay, so let's move towards the next question. The next question is, which phase of cardiac cycle ensures blood flow into the systemic circulation? बताइए, which phase of cardiac cycle ensures blood flow into the systemic circulation? Someone wants to join. निशांत का आंसर है बी. Okay, I'm repeating the question. Which phase of cardiac cycle ensures blood flow into the systemic circulation? कौन कह रहा है भाई ये ऑप्शन सी? भक्ता. भक्ता, आइसोवॉल्यूमेट्रिक रिलैक्सेशन कौन सी स्टेट थी? If you can tell me please. आइसोवॉल्यूमेट्रिक रिलैक्सेशन कौन सी स्टेट थी? It is that state when the pressure inside the ventricles is dropping. तो वो तो सिस्टेमिक सर्कुलेशन में नहीं ना ब्लड फ्लो कराने में हेल्प कर सकती. सो द आंसर टू दिस क्वेश्चन इज वेंट्रिकुलर सिस्टोल.
Okay.
Ventricular systole, which helps in the which ensures the blood flows into systemic circulation. When the ventricles, there will be systolic condition in ventricles, then also the pressure in the ventricles will be high and it will ejaculate the blood in the systemic aorta, which will lead to the circulation of blood in the whole body. Okay, so remember this point, this is a very important point. Okay, so next question is, the primary function of the AV node is to. The primary function of AV node is to. Answer to this question, everyone. Only this four or five people are active. Rest all don't they want to answer, or are they not enjoying, or something like they don't understand? What is the problem, people? Please participate. It will help you guys only. Okay. So yes, again the question is, the primary function of the AV node is to. We have options. Contract the atria, delay the conduction to the ventricles, stimulate the SA node, and depolarize the ventricular valve. Sorry. So, what is your answer on this? Okay, whoever has answered it as B, they are correct. It is the delay in conduction to the ventricles, and this is to make it in a rhythmic manner so that all the atrium and ventricles don't contract simultaneously. Okay. So the next question is, we have done all the questions in this page. Yes. The next question is, the normal blood pressure reading is. Okay, I guess all of you can answer this question. The normal blood pressure reading is. All should answer this question. Yeah, no one should get it wrong. This is a question. This is a thing we are reading from like 7th or 8th class, na? So everyone should get it right. Yes, the answer is A, 120 by 80 mm of Hg. Okay, 120 by 80 mm of Hg. Okay, the fastest conduction speed in the heart is observed in. What is the answer to this question, everyone? The fastest conduction speed in the heart is observed in. Navya, have you answered this D for question number 20?
Yes, ma'am.
Okay. Everyone, the answer to this question is the fastest conduction speed in the heart is observed in. Yes, whoever has answered or not answered, and if they find if they think that the answer is D, they are right. The answer is Purkinje fibers. Okay? The fastest conduction speed in the heart is observed in Purkinje fibers. Okay? Okay. Let's move towards the next question. Which cardiac output formula is correct? बताइए. अभी पढ़ाया मैंने आपको थोड़ी देर पहले. आप लोगों ने वीडियोस में भी पढ़ा होगा. Which cardiac formula is correct? Everyone. Yes, it is B. Heart rate into strokes volume. What is the stroke volume? The value of stroke volume? If anyone can answer.
70 ml.
70 ml. Yes, it is 70 ml. Okay, so let's move towards the next question. Which heart chamber generates the highest pressure during systole? बताइए. Which heart chamber generates the highest pressure during systole? Other people, what is your answer on this? Which heart chamber generates the highest pressure during systole? Yes, it is the left ventricle, and the maximum pressure it reaches up to is, what is the maximum pressure?
140
Sorry,
140 mmHg.
140. I'm asking in the resting condition.
रेस्टिंग कंडीशन में 80 होगा.
80 होगा. अभी
मैम
आप एक सवाल का जवाब मुझे दे के आए हैं. 120 by 80 mm of Hg. What would be the highest pressure in the left ventricle?
हाई 120.
120 mm of Hg. नहीं, it's not 120 mm of Hg. It is the diastolic pressure. ठीक है. चलिए जी. ठीक है. तो फिर उसके बाद है both the vena cava drains blood into which chamber of heart? Everyone, the question number 23 is both the vena cava drains blood into, and what are the names of these vena cavas?
Superior, inferior.
Yes, it is superior vena cava and inferior vena cava. Okay, and they drain the blood into right atrium, and the blood they are carrying is oxygenated or deoxygenated?
Deoxygenated.
Yes, it is
Deoxygenated.
deoxygenated blood. Yes, right. It is deoxygenated blood. Okay, so the next question is, which structure allows electrical signals to pass between cardiac muscle cells? बताइए. They are talking about intercalated disc. Intercalated disc में क्या structure होता है जिसकी वजह से electrical signal will pass between the cardiac muscles from one cardiac muscle to another cardiac muscle cell? बताइए. What should be the answer? Yes, it is the gap junctions. These are the gap junctions which allow this signal to pass from one cardiac muscle cell to the another cardiac muscle cell. Okay, ठीक है जी. Let's move towards the next question. We have only some questions left. Okay, so the question is, what is the function of chordae tendineae in the heart? Chordae tendineae. What is the function of this chordae tendineae? Chordae tendineae. Yes, it is to prevent the inversion of AV valves. Okay, so the function of chordae tendineae is to prevent the inversion of AV walls. ठीक है? Okay. So the next question is, during exercise the cardiac output increases primarily due to, बताइए.
हार्ट रेट.
सिर्फ हार्ट रेट
सी.
में क्या होता है? डिक्रीज होता है, इंक्रीज होता है?
इंक्रीज इन हार्ट रेट एंड स्ट्रोक वॉल्यूम.
Yes, it is due to increase in heart rate and stroke volume. इस क्वेश्चन पे किसी को कोई कंफ्यूजन है इस पॉइंट पे तो मुझे पूछ सकते हैं अभी.
मैम ये एथलीट हार्ट भी होता है ना इसके बाद से.
सॉरी.
एथलीट हार्ट का फिनोमेना यहाँ से ही स्टार्ट होता है ना.
Basically when you are in a constant, strenuous activities, you are involved in some kind of exercise or something. So in that scenario, the cardiac output increases because your body needs extra oxygen and extra nutrients. Okay. So athletes के केस में ऐसा होता है generally. Okay. So I hope this point is very clear to everyone that during exercise, cardiac output increases primarily due to increase in strokes volume and the heart rate. Okay, everyone. Okay. So next question is, what is the term for highest pressure in arteries during the cardiac cycle? Which term is used for this? Which term is used for this? Yes, it is systolic pressure. It is the systolic pressure which in resting condition reaches up to 120 mm of Hg. Okay. Okay. Next question is, which heart structure prevents blood from flowing back into the left ventricle after contraction? ये क्वेश्चन हम पहले करके आए हैं. It is just the repetition, just the language is changed. Which heart structure prevents blood from flowing back into the left ventricle after contraction? हाँ जी. Mitral valves. जागृति. Is it mitral valves? बताइए. जागृति. आपको थोड़ी कंफ्यूजन है. मैंने क्वेश्चन ध्यान से पढ़ो. जागृति. It is which heart structure prevents blood from flowing back into the left ventricle after contraction? Tricuspid valve or mitral valve? Both are the valves which are present at atrioventricular junction. But here the blood has entered the ventricle. After entering the blood into the ventricle, where will the blood go? It will go either into the aorta.
Aorta, ma'am.
Yes, either into the aorta or into the pulmonary artery, है ना? तो वहां पे कौन सी वॉल्व्स होती हैं? और यहाँ हम लेफ्ट वेंट्रिकल की बात कर रहे हैं, तो यहाँ पे कौन सा स्ट्रक्चर होगा? अयोर्टा होगी, सिस्टेमिक अयोर्टा. So the answer to this question is aortic valve, and these are semilunar valves. Okay, these are semilunar valves. You must read the question carefully. You know the answer, but sometimes read the question carefully. Okay. So the next question is, which phase of cardiac cycle occurs when the ventricles are completely closed and pressure builds without blood flow? बताइए. इस चीज पे हमने आज थोड़ी डिस्कशन भी की है. तो टेल मी द आंसर. Which phase of the cardiac cycle occurs when the ventricles are completely closed and the pressure builds without blood flow? Yes, it is isovolumetric contraction. This is that condition in which the blood pressure is increasing in the contraction is happening in the ventricles, and so that the pressure is increasing inside the ventricle. Okay. And the ventricles are completely closed because the bicuspid valves are also closed and the semilunar valves are also closed. So pressure is building inside the ventricles. Okay, the last question that we have today is, what triggers the opening of semilunar valves during the cardiac cycle? What triggers the opening of semilunar valves during the cardiac cycle? What triggers the opening of semilunar valves during cardiac cycle, everyone? Answer is, according to you, is B. Let's see all other options also. So it says atrial contraction. नहीं हो सकता क्योंकि एट्रिया के अंदर तो हमारी अयोटा वैसे भी ओपन हो ही नहीं रही है ना. So this is wrong. Another thing is higher ventricular pressure compared to arterial pressure. This is true. है ना, वेंट्रिकल में प्रेशर बढ़ेगा तो और एट्रिया में प्रेशर कम है, तभी तो क्या होगा कि सेमिलुनर वॉल्स जो है वो ओपन हो जाएंगी. ठीक, क्योंकि ब्लड विल ट्राई टू फ्लो फ्रॉम हाई प्रेशर टू लो प्रेशर. ठीक है. एसए नोड फायरिंग, एसए नोड की फायरिंग से कुछ नहीं होता, वैसे भी एसए नोड फायरिंग जो है एट्रियम्स को कॉन्ट्रैक्ट करने में हेल्प करती है. जो वेंट्रिकुलर कॉन्ट्रैक्शन होता है दैट इज बिकॉज ऑफ द कंडक्शन इन द पुरकिंजे फाइबर्स. ठीक है, एंड डिक्रीज हार्ट रेट. दिस इज नॉट एट ऑल रेलेवेंट. Okay, so these were the questions that I brought for you guys today. These are all of the answers for the questions. If you want to revise these questions, apart from that, if anyone has any other confusion regarding the last week's lecture, they can ask me. Yes, anyone has any confusion, they can ask me now. Sorry. Yes. These were the questions. I hope you guys enjoyed and try solving questions so that you guys can understand concepts.
Thank you, ma'am.
You're welcome. You're welcome. You're welcome, everyone.
Thank you, ma'am.
You're welcome, everyone. You're welcome. So, yes, anyone have any
Thank you,
other
ma'am.
confusion, they can ask me.
Thank you, ma'am.
You're welcome, Rajita. Thank You're welcome, Manisha.
Thank you, ma'am.
You're welcome, Navya. Anyone have any confusion, they can ask me. I'm still here for next 8 minutes. You can ask me the questions. Uh, okay. The things I want you guys to keep in mind is the points that I told you in this slide. Okay, these are some of the important points that you guys must remember. Okay, and also you should, for your knowledge, you should write on paper the difference between the skeleton and the cardiac muscle, apart from the structural differences. Like the refractory period of skeletal muscles is shortened than the cardiac muscles. There are intercalated discs, that is the structural point. The skeletal muscles take milliseconds to respond, while the cardiac muscles takes longer. The cardiac muscles contract in a rhythmic manner, while the skeletal muscle contract all, like it is all or none principle. Either it will contract wholly or it won't contract at all. Okay, so you guys must remember these little points. And yes, there is the gap junction or desmosomes present in the intercalated disc in the cardiac muscles for the conduction of action potential from one cell to another cell. So these are some little points that you guys must remember. Yes, the T tubules in cardiac muscles are 5 times larger than the ones that are present in skeletal muscles. So this is also you should remember. There's this plateau phase in the contraction of the cardiac muscle, which is not there in case of the skeletal muscles. This plateau phase is because of the calcium ions. So this is also a point to remember. So these are some of the little points that you must remember while studying these contraction and other things. Okay. Any other thing you guys want to ask me, you can ask. Anji Bhajni, you want to ask something?
मैम
Yes, Bhajni, बोलिए.
24 आंसर
I can't hear you, Bhajni. Can you please speak a bit louder?
She asking 24 answer.
24 का answer. Okay, I'll just reach to the 24th question. Is 19th. Okay, which structure allows electrical signals to pass between cardiac muscle cells? It is gap junctions, Bhajni. These are the gap junctions which allow the action potential, or we would say the electrical signals, to pass. These are the gap junctions. You see here, desmosomes, gap junction. These are the gap junctions. These are the gap junctions. These gap junctions allow the action potential or the electrical signals to reach from one cell to another cell. Okay. Any other thing? If anyone wants to ask, they can ask me. I guess someone raised their hand. No? Okay. I hope today's lecture was clear to you all.
Yes, ma'am.
You have any kind of confusion in the
yes
questions
मैम
in the assignment?
No, ma'am.
Okay, one more thing that you guys must remember is the difference in the calcium ion concentration. There was no such thing we discussed in the skeletal muscles, but here in cardiac muscles we are discussing that thing because there is a huge difference between the calcium ion concentration across the cell membrane. That is nearly 1000 to 10,000 folds. In the inside, the calcium ion concentration is around 0.0001 millimolar, while outside it is 1.2 millimolar. So it is nearly 1000 to 10,000 folds is the difference in calcium ion concentration. That is why we need a calcium ion rush from outside of the cell to inside of the cell so that the conduction that contraction can happen properly. Here the sarcoplasmic reticulum also are not much developed as compared to the skeletal muscle, so the calcium ion from outside is a must condition for contraction in case of the cardiac muscle. This is an important thing, you guys must remember this. Okay. Okay, if there is no more questions you guys have, you can leave the meeting. Yes, Bhakta, you want to ask something?
No, ma'am.
Okay. You guys can leave the meeting if there are no more questions.
Thank you once again, your class, ma'am.
You're welcome. I hope I'm pronouncing your name to be correct. Everyone, you can leave the meeting now if you don't have any more questions. Suresh Balaji, you want to ask something? He's left the meeting. Okay, everyone, have a very good night. Thank you for attending the lecture.
Good night, ma'am. Thank you.